right, we're back in the Morning Brew with Friends with our friend Bridget Wilson from UNM Cancer Center, and this is cool. This is the new show, and it's called Inventory of Light, and I dig this, because this is, you know, most of us, we think of biology, we don't get to see the cells, do we?
You don't. They are very small.
Right.
So you need special instruments to see them.
And you guys have them, and as you've been looking through these special instruments, you've just been seeing some beautiful stuff.
That's right. We at the UNM Cancer Center, we take advantage of some really fantastic technologies. We also use microscopes at San Diego National Labs, and we use really powerful computing.
Are these like electron scanning microscopes? And
I use the electron microscope quite a bit in my work.
What do you research?
Pardon?
What do you research?
What do I research? Well, I'm a cell biologist, a biochemist, and a systems biologist. So the cell biologist in me thinks about the very smallest unit of life. That's the cell. Inside of it, all its parts. And then the systems biologist part of me thinks about how that fits into the whole, in kind of a holistic way, how that fits into the system of the body, and how those cells in specific locations do their job.
But then you also have this artistic mind that
That's
saw
right.
something in this too, right?
Well, when you look in the microscopes, we use one of the reasons the show is called Inventory of Light is because we use fluorescence microscopy quite a lot. So we use the power of fluorescent tags to look, and they're very colorful. They come in all colors of the rainbow.
Like this
here
one
we
here?
go. This particular image.
What is that of?
So this is an image of it's a rather an unfortunate situation. This is an image of those green little spots you see are each leukemia cells that have invaded a brain.
Wow.
That blue, sort of purple blue border that you see, and the red in the inside is the vessel, the red cells in the inside of the vessel, and so what you see
and you dye the cells, and then with special light, they fluoresce.
Those cells have been engineered to fluoresce green. And the other cells are dyed. And so then you can see the whole architecture of the tissue. And in that particular case, we're trying to figure out how we can better target the leukemia cells that are in the brain.
Well, can't you just say everything that's green, I want out of here?
Well, it doesn't work that way. Otherwise, you might end up, damaging the brain. And this happens to be a childhood leukemia. So you need to approach it with thinking about the toxicity of the drugs you give the children.
Right.
And
so you over the years, you've been seeing all of these amazing photos and these amazing images. And then how did you take that leap from saying, I see these every day in my lab and I want to share them with the world? How did you guys get to that point?
Well, one of our scientists, our Los Alamos colleagues, had experience with the Santa Fe complex where they had been giving public talks. And it was really well attended. Santa Fe's quite an intellectual and exploratory kind of community.
Right, it's very continental that way. I love it.
And so we decided that we would have an art show and public talks, and that was about 6 years ago. We moved to the 333 Montezuma Gallery, and now for the first time we're at the Gerald Peters Projects Gallery.
And that's one of the largest in Santa Fe. It's
beautiful,
an amazing beautiful spot.
gallery.
And the work that I'm assuming is for sale too.
There is work for sale there. The scientific art probably couldn't be negotiated. We have had, last year there was art for sale. But one of the things that's interesting is this has kind of taken the country by storm, because we were a pioneer in having this kind of event, but now you can go to the Dulles Airport, to the Chicago Airport, to the San Diego Airport.
And see the similar kind of work.
And see these pieces of art from all over the world.
What's one of your favorite things that you've ever found? What's one of the favorite pieces you've seen?
Well,
do you look at it in terms of aesthetics or what is actually on the slide that you're looking at?
So we do both. When we're choosing art for the show, we're thinking aesthetics.
Right.
But when we are looking at it for informational purposes, we're looking at how molecules traffic, and that can be really important, because how things get around in the cell if it's aberrant, it can cause the cell to proliferate too fast, do all the things that are a problem in cancer. I also study allergy, so that can trigger all the events that cause you to sniffle this spring season.
Okay, that's a problem that all of us here at the Morning Brew have. We're waking up early and things are pollinating, and we're all sniffling and sneezing. Do you have any advice on the allergy front?
Well, on the allergy front, there are some new, very new, interesting findings that are coming out. So many women will remember when their babies are born, they go see the pediatrician. They've been advised to stay away from peanuts from day one. And there's a brand new study that came out from the United Kingdom that suggests that might not be the best strategy. Little tiny doses as infants. So that's something you might want to talk to your allergist about.
That's interesting. Yeah.
Don't take it from me. Take it from your allergist.
So the show it goes up this weekend, right? March 27th.
The show goes up Friday, and the show will continue being up most of the month of April. But the big events this weekend are Friday, Saturday night receptions. You can see all the scientific art, and then you can also see there's 8 artists that have come from all over the country whose work is inspired by science.
Oh, cool.
And so it's really quite innovative.
And we're looking at this, as lay people who don't know anything about science. And I'm looking at that and just thinking this is beautiful, but I'm sure there's scientific, studies and results that have come out of this. I mean, what are some of the things that have come out of this work, some of the research that's come out of this work?
So there is a display of work on going in the allergy theme, because one of the public talks is on allergy, and that is you get to look at scanning electron micrographs of pollens at their finest level. That's what's in the air, making everyone feel
Right,
bad.
that's the stuff that makes us sneeze.
And then we have a series on cancer, and so you can look, just as we looked just a few moments ago, at leukemia in its place. There are images of lung cancer and melanoma, but they're in an artful sort of way, and scientists are present to talk to you.
Great.
So that's what's really great.
And we have a video you guys
We do
shot
have
of
several
this.
videos.
I think we got it queued up here. We can watch that. And what is the video has?
Oh, now this video is one of the artists that's coming in that the Gerald Peters Gallery has brought in. And it really is a fantastic illustration of a system. So this network is an interwoven network of microcontrollers and ambient sensors. But really, these fluttering leaves, they look like butterflies.
Yeah.
It's a sculptural system that responds to the audience. So this is one of the interactive.
So if I walk up to it, the leaves will flicker?
The leaves will change.
That's cool.
And it's an illustration of complexity. The artist talks about the process of nonlinear dynamics, really how you drop a pebble in the water and ripple effects occur. But in this case, in a nonlinear fashion.
Very
so
cool.
I can't wait to see this,
Yeah, it looks
particular
great.
one.
Very cool. And I love art that you can kind of interact with and play with, too.
This is...
That's not the Gerald Peters Gallery, though.
This is going to be displayed at the Gerald Peters
Yeah.
Gallery. So this is not its original installation, but you will see that here this weekend.
Very cool. That's very cool. So that's how you have to look at these systems. You look at looking at the one leaf, you have to look at the leaf and then the whole system.
That's correct.
In your research.
That's exactly true.
Very cool. So in your research, what are you studying now? What's the big thing that you're looking at diving into right now?
So one of the things we study in our Systems Biology Center is the field of cancer right now is undergoing quite a dramatic change, because now we have what you would call targeted drugs or tailor-made drugs.
And there's a lot of genetic drugs.
And those are based. These drug treatments are based upon knowledge about the individual person's cancer, the genetics of the cancer, and the defects that come after those mutations, that are linked to those mutations. And one of the challenges now is to determine, out of this larger choices of drug therapies, what, in a precision kind of way, what that individual patient should go on. And to understand that, you need to think about how the workings of the cell. So that's our
dive.
And so we'd like to have very good, reliable, predictive models of how personalized response will occur, and then that will guide better choices.
Excellent. Well, just last question quickly. The show opens this week. What's your favorite piece in the show that you want to have everybody see?
Oh, my favorite piece is one by Thomas Derink. He's one of our colleagues at University of California. It's pretty stunning.
And what's it an image of?
It is an image of the brain in multicolors. It's
Very
quite
cool.
fantastic.
You can see the entire brain.
That's right.
Excellent.
I thought we would see it, but
All
we
right.
didn't.
Well, we'll be back in a couple of seconds. We got our buddy Steven, the Curious Townie. Steve Westman's going to be here in just a couple of seconds on the Morning Brew with Friends.